Hey, I'd like to welcome you to another episode of Mission Matters. My name is Adam Torres, and if you'd like to apply to be a guest on the show, just head on over to missionmatters.com and click on Be Our Guest to Apply. All right, today's a very special episode. We have Anil Diwan on the line, and he is the founder over NanoViricides Inc. And this particular interview is part of our DealFlow Discovery Conference series, where we bring you the best of those that are attending and participating in the DealFlow Discovery Conference that's taking place at the Borgata in Atlantic City, New Jersey. I've been before. It's a beautiful, beautiful casino and area. You definitely want to check this out. And you can get tickets and learn more at two different places, at the DealFlowEvents.com website or DealFlow Discovery Conference website as well. So you can go to either of those, and Anil will be there in attendance. So you'll be able to ask him some questions for yourself as well if you choose to attend. So definitely go grab some tickets. Anil, welcome to the show. Good to have you here.
Thank you very much. It's great to see you.
All right. So we got a lot to talk about today. I want to get into what you're doing over at NanoViricides Inc. For everybody that's watching, that trades under the NYSE ticker NNVC. So that's Victor Charlie. And check out NanoViricides. So we got a lot to talk about today, Anil. But before we get into the company, what you're doing, NV387, we got a lot to talk about. Let's get into your mission. So we'll start this episode the way that we start them all, with what we like to call our Mission Matters Minute. So Anil, at Mission Matters, our aim and our goal is to amplify stories for entrepreneurs, for executives, for experts. That's what we do. Anil, what mission matters to you?
So NanoViricides was founded with the mission of developing revolutionary antiviral therapies. If today you get sick with a viral infection, you go to the doctor. First of all, you pretty much for 2 days, you will stay in bed. Then you will go to the doctor. And the doctor says, oh, it's a viral infection, nothing to do, go back. Right? We want to change that, right? Yeah. And the reason we are saying that it's revolutionary is because this is what ampicillin, amoxicillin, these penicillin type of drugs did for bacterial infections. Before them, people were dying of bacterial infections. When those penicillin type of antibacterials came into being, now when you go to a doctor and you have a sore throat, they say, oh, it's a bacterial infection, strep or something like that. Here's the prescription. And then you go take the 7 days course and you're fine. Right? So that's the difference between how we treat viral infections today versus how we treat bacterial infections today. And that kind of treatment is called empiric therapy. And so we are developing, our first mission is to develop an empiric therapy for acute viral infections, typically the respiratory viral infections like RSV, COVID, influenza, and all those other things that you always seasonally get. Secondly, there are hundreds and thousands of viruses that infect humans. You can't develop a vaccine for every single one, and you can't force that into pediatric vaccination program for every single vaccine, right? We already have huge number of vaccines in the pediatric program, and most countries in the world are trying to cut down the number of vaccines that are given in the pediatric programs. And US Secretary has taken a lead on that. So that's where we are. Our mission is to develop revolutionary viral therapies.
Yeah. How did you, it's a big mission. It's a big, big mission. And you're trying to affect and help lots of people. How'd you get into this business? Like, where'd that start? How'd you pick this specialty? Like, knowing that you were going to go down this route?
That goes to a very long ago. Something happened. There was a first gene therapy trial where they had used adenovirus as the vector for the gene therapy, and the patient died. So I was talking to a research director of a big company, and I said, why are they using a virus as the vector for genes to be introduced? And when they're doing that, obviously the virus can cause an infection, something can happen, some other effects can happen. So why are they doing that? And he said, it is impossible to do that without using a virus. I said, why? And so he explained to me the complications of where things have to go, and so on and so forth. So I said, no, I'm a chemical engineer, by the way. My initial training, bachelor's in chemical engineering from IIT Bombay in India. Then a PhD in biochemical engineering from Rice University. Then I did biomedical engineering, and then I did virology work. And after that, I started my own company. That's when this discussion happened. And
so you didn't have a random opinion like me, where I'm like, no, I don't think that. I don't know.
So no. He used the word impossible.
Yeah, you didn't like that. I know. I have a feeling as soon as you heard that, you're like, you just Perked up a little, like, wait a minute, hold on. That's a strong word.
Yes.
Continue, please. I just couldn't let that go.
Right. So that word doesn't fit in,
Yeah.
So I said, okay, let me try to think about it. And I have polymer background as well. So I developed some concepts about how we can do something. Viruses are distinctly different from our cells. So it is easier to target viruses.
Yeah.
And the knowledge that was there at that time for being able to target viruses was only for influenza and HIV. Now there is a whole lot of things have happened, and we can practically target almost any virus that causes human infection with our technology, this NanoViricide technology. So that is where the whole thing started. And then we were talking to somebody who had worked with early HIV cases. And that medical doctor, Dr. Eugene Seymour, the first time I met him at a kosher restaurant in Manhattan.
Yeah.
And I was very late because there was a United Nations meeting going on at that time, which I didn't know about. And I decided to drive because I thought driving will take me faster. So he was walking out of the restaurant and I just grabbed him back in. And because of that, I just ran from my car and I didn't take my briefcase. So I didn't have a paper to write on or a pen. I took the napkin there. I pulled his pen and drew some structures on the paper, and I said, this is what I want to do for virology. This is going to be a drug for viruses.
Wow.
Yeah. He looked at me and said, you must be crazy. So that initiated a lot of scientific discussions, scientific due diligence from his people, friends and faculties of different universities. And they all said, this guy probably is crazy, but he may be onto something, and he definitely is smart. Okay. Yeah. So that definitely is smart is what stuck with him. He said, okay, I want to do this. Wow. So that's how NanoViricides was founded, with Dr. Seymour as the co-founder and me together. And since then, we have improved the technology tremendously. This I'm talking about 2005 when we founded the company. And now it is 2026. And along the way, around 2010 or so, we realized we need strong controls. We need capability to be able to manufacture the same drug again and again the same way, meaning it has to be similar and it has to be able to work the same way. So because of that, we took upon ourselves to build a facility for manufacture also. And that's what we have done. In 2015, we moved to the new campus. We developed a CGMP capable manufacturing facility there, CGLP capable laboratory inside. So we are now soup to nuts complete pharmaceutical company. It's called a vertically integrated pharma company. So right from concept, discovery, all that modeling, molecular modeling and computer modeling and everything, to synthesis, developing synthesis pathways, scale up of manufacture, and then GMP manufacture. And then during the COVID, we also did actual formulation of the final drugs. So everything now formulated drug is being sent from our facility. United States based manufacture of never before done type of nanomedicines of a completely different architecture. So, that's a first in a world, essentially.
Yeah. It's amazing. I want you to talk a little bit about the pipeline and what you're working on now. Maybe start with NV387.
Right. So NV387, like I said, the mission for an empiric antiviral, just like the bacterials, which was our first purpose, that NV387 satisfies. We developed NV387 very vehemently during the COVID pandemic. So March 2020, we already had indications that it was working against coronaviruses. And then by January 2021, we had finished safety toxicology studies, the required studies at that time. And then we got stuck with, because we never had regulatory background. We are chemists, chemical engineers and formulation scientists, but no regulatory affairs here at that time. So, and because the whole way that the government funding went and the whole mechanics of that time went, the regulatory affairs people became completely unavailable. Regulatory sites for clinical trials in the United States became completely unavailable, especially to small companies like us. So we went to India. Eventually, we were able to make a representation there and get a site there, and we were able to do clinical trials there. the problem that happened is that by the time we finished our phase 1 clinical study, which had a main component was the healthy subjects exposure and study safety and tolerability. The second part was to be able to do the same thing in patients of COVID. The problem that happened was by the time we finished the healthy subject study, there were no patients available. We did actually for 2 weeks, we kept testing with RT-PCR, spent a lot of money on that, but nobody was positive and we couldn't continue that part. So we had to close the clinical trial at that. So NV387 was shown to be safe, well tolerated at very high dosages as well, more than what we need to use in the clinic. So that was done. And so that is when we started checking, okay, the reason NV387 we expected to work against many viruses, and it is expected to work against maybe 95% of respiratory viruses. The reason for that is because the way we did this is completely different from what people do. What people do is they attack the virus once it's inside the cell and it is trying to make copies. Now, when they are doing that, they are doing something that affects the machinery of the virus or the machinery of the cell. And those chemicals that they do tend to be not very safe. To some extent, they are toxic. And most of them, the safety toxicology limit and the tolerability limit and the efficacy limits, there is not that much distinction. So you cannot you may be able to go to double the dose of the clinical level that gets approved, but you may not be able to go to 4 times the dose level. So there is a limitation automatically in how effective they can be. So with ours, what we decide to do is there is a different thing. The viruses bind to something in the human system. And that is where they get a handle on how they are going to go into the cell and walk into the cell and then hijack the cellular machinery, make copies of themselves. So that very first step called infection, technically it is called reinfection, because the first infection is when the virus is aspirated into your body, either from something that you ate or in a wound or by nasal aspiration, respiration. So that's the first step. That's called infection. And then when the virus is made into the body in the first infected cell and goes out, starts infecting and starts spreading, that is called reinfection cycle. So we decided to affect that cycle, and the reason for doing that is that no matter the virus mutates, it binds the same exact points on the same exact proteins or receptors in the body that it was going to bind to. So the idea is to get a drug that the virus cannot escape. And how would you do that? You have to have something that doesn't change, right? So every single thing that people have done to date, trying to create broad spectrum drug candidates, they have always failed because of this thing that the virus mutates. In COVID, we saw that very effectively. Everybody became aware of it because the antibodies that developed with great fanfare, with billions of dollars each antibody, they failed within 3 months. As soon as they were introduced, the virus mutated away from them. Vaccines, the same thing happened. 6 months later, a new vaccine had to be introduced. So that is the problem with viruses. They can mutate very rapidly. They can change very rapidly. And I'm using the word mutate in a very general sense. The technological things that the viruses do to change themselves there are many different ways that they do. But we'll collapse all of them into the word mutate. Okay? So to do that, so viruses basically use a specific signature in the human side, and that is what that will bind to. And for example, some 95% of viruses bind to what is known as heparan sulfate proteoglycans. So that's heparan sulfate is a polysaccharide, and it's called proteoglycan because polysaccharide is called a glycan, and it is attached to a protein somewhere. So because of that, it becomes proteoglycan. When it binds to that, it concentrates there right next to the cell, and because it has concentrated many virus particles together, now they have the ability to attack the cell by binding to another receptor on the cell surface to be able to go inside. So that second receptor is called the cognate receptor. So in the case of SARS-CoV-2, for example, the ACE2 receptor that everybody has talked about, that is the cognate receptor. But all coronaviruses bind to heparan sulfate first. All influenza viruses also first bind to heparan sulfate proteoglycans. So there are some 95% of viruses that bind to first to heparan sulfate. So what we decided to do during the pandemic is that, okay, we will copy the heparan sulfate. Now, if we took the same thing, the heparan sulfate itself, then we don't have an advantage against what is in the body, right? We have to have something better, stronger that will capture the virus.
Yeah.
So it's like the Venus flytrap, right? So it has to be something better than the other thing that is around. So because of that, we did a lot of molecular modeling. We did a lot of work initially, and we came up with some chemical structures. We attached them to the polymer backbone that we had developed. The polymer backbone basically makes it look like a cell surface to the virus. And the other thing in our nanomedicines, what is very different from what other people do, most every nanomedicine that has gone in cancer related research, for example, they have been particles, and so most people say nanoparticle instead of nanomedicine. That is different. What we do is not a particulate matter. What we do is a soluble material. It's almost like soap micelle, but basically we do what is a micellar solutions. These nanomaterials that we develop, they remain in solution. And so that gives it a lot of advantages. The other thing that we did is that this material is not in a fixed shape. Particle will be in a fixed shape. If you have one particle, this is your nanoparticle, let us say, and this is your virus particle. They bind to each other, they bind tangentially. It doesn't give much of a binding, strong binding. So if you have two heavy balls that bind tangentially, you know that they separate easily, right? So what we did is that this is your nanomedicine. Once it binds the virus particle, it has a lot of additional fingers that it starts binding with, and it covers the virus particle. In the process of doing that, it can destroy the virus particle. And we have seen that in electron micrography. We have demonstrated that by fluorescence related studies. So we have now developed a technology that is a shape shifting technology that goes after the virus and discombobulates it. The virus can no longer infect cells once it has done that. Right?
Man,
and that's very different from what anybody else is doing.
That's exciting, Anil. I'm not going to lie. It sounds very sci-fi to me. I tracked everything you said. It's amazing. It's amazing. Like like just the thought process behind how you're attacking this, and that it actually, how far are you with this particular model? Like, is this in
I said, phase 1 was completed with NV387. Right?
Yes.
And now we are going into phase 2. So along the side, we were testing it against many different viruses. We tested against coronaviruses, of course. Yeah. After that, we did animal studies against influenza H3N2, the one that is causing havoc right now. The drug works very well against that in animal studies. It works way superior to Tamiflu, Xofluza, the drugs that are being talked about right now. That, and those drugs are susceptible to viral mutations. Once you deploy those drugs, within 3 months, you are going to have mutants, that kind of drugs. So here we have something that is resistant to that. The viruses would not mutate against, and it is very effective. We did against RSV, and it was highly effective. In fact, it cured mice of RSV. Okay. Now, a lot of people cure mice of cancer also, but nobody has cured mice of RSV, and we did that. So I'm trying to say this in a fashion because curing mice is easier than curing humans. A lot more things are involved in curing humans. So we have some steps to go, right? We even worked against Mpox. In 2022, there was a Mpox scare. And in fact, Mpox has become endemic in United States. And fortunately, that particular virus in United States is clade 2, which is less vigorous, less infectious, and spreads less rapidly. And because of that, it remained limited to a community, basically gay community. And so it has not, people have not become too aware of it. The drug that we developed, NV387, worked against that also in animal models. Mpox last year, 2024 August, created a huge emergency in Africa. It's still continuing. It has started dwindling down now. But since that August 2024 news line came out, press release came out from WHO, we have been trying to get into clinical trials for Mpox. Nearly 6 months, nobody was even listening to us. Eventually, we made some roads in there, inroads into there. We were able to get into DRC, Democratic Republic of Congo, and set up a team. And now we have pretty much completed all the requirements of the clinical trial application was completed. It was sent out, and we are just waiting for some other small things to be completed. After that, we will be able to start getting dosing into humans for phase 2 in Mpox. Given a chance by the regulatory agency in Africa that, okay, the data that we gave to them is very compelling.
Yeah.
So,
it's good. Well, Anil, I wish you would have been my biology teacher growing up. I'll say that. This has been, you taught me a lot today. Like, to be honest, you taught me a lot about what was going on and you explained it very clearly. So I learned a lot. I'm sure my audience did as well. So how do people follow the company? How do they follow the company? How do they follow the journey?
So we do press releases often, and we also have our own website, NanoViricides.com, and our press releases go onto that. And that's pretty much all. We don't do direct email, or we don't have a roster where we can send emails to people on list and things like that. We will get into that. Right now, it's a small company, and we try to put all our money into where it is needed to go is drug development.
Amazing. For everybody listening, just so you know, we'll definitely put some links in the show notes of the website, all that good stuff. And as mentioned before, so Anil and his team will be over at the DealFlow Discovery Conference at the Borgata in Atlantic City, New Jersey. That's going to be January 28th and 29th. Go out there, see him, and see all of the others that have been on this program for the DealFlow Discovery Conference series. And speaking of the audience, if this is your first time with Mission Matters and you haven't done it yet, hit that subscribe or follow button. This is a daily show. Yes, you heard me correct. Daily, each and every day, we're bringing you new content, new ideas, and hopefully new inspiration to help you along the way in your journey as well. So again, hit that subscribe or follow button. And Anil, man, thank you for coming on the show today. I will continue to be following this company and your journey. So thanks again for coming on.
Thank you very much. Appreciate it.